Blanching is an important part of processing fruits and vegetables. So what is blanching and how do we blanch foods? We'll answer these questions in this presentation. Since enzymes are involved, we'll explain what enzymes are and how they work. Then we'll look at enzymatic browning and a couple of examples of enzymes, namely catalase and peroxidase. We'll wrap up by looking at the blanching of food products. So just what is blanching? Simply stated, blanching is a process in which fruits or vegetables are submerged in boiling water for a given period of time. They are then removed from the boiling water and plunged into or sprayed with cold water to stop the blanching process. So why do we blanch food? Blanching accomplishes two main objectives. First, it essentially pasteurizes the surfaces of the fruits and vegetables and reduces the population of microorganisms that may be growing there. Second, it deactivates enzymes which are naturally present in the fruit or vegetables and can lead to the loss of quality and spoilage. Blanching does not really cook the food. It provides sufficient time at a suitably high temperature to effectively kill the surface microorganisms and deactivate or denature the enzymes that are present. Most fruits and vegetables should be blanched before they are frozen for storage, and as well they should also be blanched prior to drying. Radishes are a good example of this. What is an enzyme? An enzyme is a protein molecule that has bioactivity. This means that it is biologically active and can act to speed up chemical reactions that would otherwise go very slowly or perhaps not at all. As biological catalysts, they are specific, meaning that they promote only certain reactions and no others. One particular model used to describe how enzymes behave is called the induced fit theory of specificity. This is just a fancy way of comparing an enzyme to a lock and key situation. Enzymes have active sites which promote the desired reactions. Enzymes take a starting material called the substrate and convert it to reaction products. Generally, enzymes are named by taking the name of the substrate material and adding the ending ase to it. For example, if we have an enzyme that reacts with peroxide, it would be called peroxidase. Consider the enzyme invertase. It was named before the naming convention was adopted, and it is involved in the conversion of sucrose to glucose and fructose. So we'll show that reaction with an arrow and indicate the enzyme above the arrow in the reaction sequence. If invertase is placed in a solution of lactose and sucrose, it will only react with the sucrose, even though both sugars are disaccharides and have the same molecular weight. Therefore, invertase is specific for the molecule sucrose. Here we have a sucrose molecule represented by the white block in this diagram. The enzyme molecule is represented by the black block. The two red asterisks indicate that these are active sites, and you will notice how the sucrose molecule is hovering just over the enzyme molecule. Now we'll also have some water molecules involved in this process as well, since this reaction will take place in solution. The sucrose molecule will then align with the active sites on the enzyme molecule and will actually couple with the enzyme molecule. Then the water molecule will move into place and it will hydrolyze the bond between the glucose and the fructose, which make up the sucrose molecule. At this point, the glucose and fructose molecules will leave the active sites, as indicated by the arrows going to the upper left and to the upper right, and the enzyme molecule remains unchanged. The active sites continue to be available for the conversion of more sucrose to glucose and fructose. Enzymatic reactions are affected by a number of factors. The first one is pH, or the acidity of the solution. If a solution is too acidic or not acidic enough, the enzymatic reaction will not proceed, or it may proceed more slowly than it optimally would at the correct pH or correct acidity. Temperature is another thing. Many of the enzymatic reactions prefer what we call physiological temperature, or about 37 degrees Celsius, which is normal human body temperature. The substrate concentration will also have an impact on the rate of an enzymatic reaction. Since the more substrate molecules are present in solution, the higher the turnover rate will be from the substrate to the reaction products. Because they are proteins, they can be deactivated in the same way as other proteins are denatured. One of the enzymes of primary concern in blanching is polyphenol oxidase. Polyphenol oxidase, or PPO as it is often abbreviated, is an enzyme that catalyzes the formation of brown or black pigments in fruits and vegetables as they age. This process is referred to as enzymatic browning. Cauliflower is particularly susceptible to enzymatic browning. Here you see a fresh cauliflower with creamy white florets. However, with the passage of time and in the presence of polyphenol oxidase, this can change, and we will see the development of brown or purple or black pigmentation in the florets of the cauliflower. So the fresh cauliflower then can become browned over time in the presence of polyphenol oxidase. Let's take a look at the enzyme catalase. Catalase enzymes catalyze the conversion of peroxides to water and oxygen, as shown by this equation. I've taken the liberty of balancing both sides of the equation so that we have the correct number of hydrogen and oxygen atoms present on both sides. This reaction is catalyzed by the presence of catalase, and the oxygen will be given off as a gas in the form of bubbles. Note how 2 molecules of peroxide are converted to 2 molecules of water and 1 molecule of oxygen, which is given off as a gas. Catalase is found in certain meat tissue but not in plants. However, it's a good example to show how enzymes work. Pouring a weak hydrogen peroxide solution over some beef liver will give us a violent reaction as oxygen bubbles form. The reaction of catalase and hydrogen peroxide is shown in this pictorial representation, where we have a piece of calf's liver and we're going to add some hydrogen peroxide to it, and what ends up happening is that we get the violent evolution of oxygen bubbles, which shows up here in this photograph as a white foam, and you can see the piece of calf's liver just below the surface of the foam in the center of the photograph. If we take a sample of the beef liver and submerge it in boiling water for several minutes, we will deactivate the catalase enzyme. This is because the input of heat changes the structure of the enzyme so that it is no longer biologically active. The reaction of heated beef liver and hydrogen peroxide is shown here. Notice how the beef liver has changed color due to its heat treatment, and then when we add the hydrogen peroxide, nothing will happen. Peroxidase is another enzyme that I would like to take a look at. Peroxidase enzymes also catalyze the conversion of peroxides to water and oxygen, but this time they do it in plants. The reaction is exactly the same as we saw before with the catalase. However, the enzyme in this reaction is peroxidase. The question that now arises is, why would there be peroxidase enzymes in plants? Peroxide molecules are formed as byproducts of a plant's metabolic processes, such as respiration. If the concentration of peroxide was allowed to build up, it would be harmful to the plant. The plant, therefore, produces peroxidase enzymes to consume the peroxide as it is formed. We will now use the peroxidase enzyme as an example to show how blanching works. We can then extend this learning to enzymes present in plants that cause degradation, such as the polyphenol oxidase we saw in cauliflower. For our example, we will take a slice of raw turnip, cut it into smaller pieces, and place them on a saucer or in a bowl. Then we will pour some hydrogen peroxide solution over these pieces, using 3% hydrogen peroxide like you can buy at the drugstore. After a short time, we would expect to see oxygen bubbles forming around the edges of the turnip. So here we see raw turnip slices on the left, and when we add hydrogen peroxide to them, we see the evolution of small bubbles which indicate that oxygen is being given off. We have now shown that there is peroxidase present in the turnip. We did not add any enzymes, so the bubbles of oxygen must have been generated by enzymes within the turnip itself. Now we will see what happens when we blanch a slice of turnip. Then we will place a slice of turnip in a heat-resistant container, such as a coffee mug, and pour enough boiling water into the mug to completely cover the turnip slice. In the case here, I found that it was necessary to fill the mug about half full. We'll stir the contents of the mug with a fork and replace the water in the mug with fresh boiling water after about 1.5 minutes. This will ensure that the temperature is kept high and that it does not fall below the temperature required for the blanching of the turnip. After about 3 minutes, we will pour the water out of the mug and run cold water over the turnip slice to cool it down and stop the blanching process. Then we'll cut the slice of blanched turnip into strips and place these strips in a clean saucer. We'll pour some 3% hydrogen peroxide solution over the blanched turnip and watch to see what happens. So here we see the slices of blanched turnip in the photograph on the left. We'll pour some hydrogen peroxide, H2O2, over these slices, and even after 2 minutes, we don't see any bubbles forming, and the slices on the right of the photograph are shown there with no bubbles around them. If the blanching was done properly, the peroxidase enzyme should have been deactivated. No bubbles should have been observed coming from the edges of the blanched turnip strips. And that's what we saw. Here we see the raw turnip with the hydrogen peroxide, and in the photograph on the right, we see the blanched turnip strips in hydrogen peroxide, and the difference is quite obvious. So in summary, we can use our experience with peroxidase to show that enzymes can be deactivated by a heat treatment process which we call blanching. If peroxidase is deactivated by heating, then we would reasonably expect other enzymes to behave in a similar manner. This would indicate that blanching should be effective in deactivating degradative enzymes such as polyphenol oxidase. In conclusion, blanching works. Blanching should be done before drying or freezing any fruits and vegetables where there is a risk of enzymatically induced degradation or spoilage. There are probably some other conclusions, and I would ask you to think about those on your own. Thank you.